Good morning, dear students. I am Dr. Anmol Kumar, Assistant Professor, Department of Biotechnology. Today, I will take the topic of tissue processing steps. This tissue processing steps also belongs to first unit of the introduction of histopathology and basic steps. In last video lecture, I have discussed about the overview of histopathology. In that topics, I have discussed about the various types of biopsy samples and how to differentiate the different types of biopsy samples. Then I also discussed in the last section of that video is how to handle the specimen and gross examination, right? Then after the gross examination, then finally tissue need to be sectioned. So that section of the tissue to be done, before that one tissue need to be processed, right? Then why tissue need to be processed, you should know. Because tissue, as you know, usually very small in size and it is very soft, so directly you cannot use for the sectioning under the microtome. So you need to provide some external support and rigidity to the sample, then only it can be cut under the microtome. That's why you need to process the tissue. So tissue processing is actually a procedure between the tissue fixation and the embedding steps. And I already said the main aim of the tissue processing is to embed the tissue in the solid medium that provide the rigidity to the samples. Then it can be cut easily using the microtome, right? So there are usually two types of tissues are there, soft tissue and hard tissue. If tissue is soft, then no need to go for the extra steps which need to be done in case of hard tissue. If you are using hard tissue, then you need to use the extra step. This is called the decalcification of the calcium. It is present into the tissue. So in case of hard tissue, you have to do extra step of the decalcification. So I will explain some more detail about the decalcification. Then decalcification, as the name implies that the decalcification is the removal of the calcium salts from the hard tissue like bones, tissue. In some cases calcium also deposited in tissue. So you need to remove the calcium before sectioning, because if you not remove the calcium, then it may create the problem to the blades which has been used in case of microtome. As you know, blade is very costly. If calcium will not remove, then it will spoil the blade, and also you will not get the good thin paraffin section. Then your morphology of the structure tissue will be changed and you will not get the good section, and finally your results will not come as per your expected results. So you need to decalcify the hard tissue. So there are various types of reagents are there, strong mineral acid based and weak organic acid and chelating agent. So whatever reagents you are using is depend upon the types of the tissue and what process you are following. So if you are using this type of reagent, they need to follow the four main criteria. First, they should remove the complete calcium from the tissue. Second, it should not take much time. It should complete within a stipulated time. Next, it should not create any problem in the staining process. And further, it should not create any damage to the tissue. So these four criteria should be followed if you are using any type of reagent used for the decalcification. So I will explain some decalcification reagent used in the tissue decalcification. So strong mineral acid mean it will remove the calcium very fast. So in this case you should be very careful and you should detect the appropriate end point. So if you not determine the appropriate end point, then in that case what will happen, the tissue will get over decalcified, then the morphology of the tissue will be changed, and moreover it will also create a problem to the nuclear staining. So you should be very careful when you are using the strong mineral acid based decalcification reagent. Mostly it will be used for the hard cortical bones. So these are the some chemical used for the decalcification. The nitric acid HCl is the most commonly used decalcification reagent under the strong mineral acids. Some other reagent also there, so that will depend upon the tissue types. Then I will discuss about the next weak organic acid. So it is mostly used for the bone marrow and soft tissue where the calcium deposition is less. So in that case, you can use the weak organic acid types decalcification reagent, and it is very slower and gentler than the strong acid. So no need to bother about the tissue damage, but and it also not create that much problem to the nuclear standing. So it is better than the strong acid, but it will take some more time compared to the strong acid. So in this case, there are formic acid in concentration of 10%. It is the most commonly used in case of weak organic acid. So it is all rounder decalcify, you can say like that. And there are another decalcification reagents are there. So in all the cases, formic acid is common, only some extra additive is added, like sodium citrate, sodium formate, in some cases is formalin. So in case of a Goodling reagent, it will provide both the option fixation as well as decalcification. This is extra advantage using this Goodling and Stewart reagent. And last type of the decalcification reagent, the chelating agent. In that chelating agent, usually you are using the EDTA. EDTA is a chelating agent. It will make a non-ionizable soluble complex, and it will slowly reduce the size of the calcium and slowly remove the calcium from the tissue. It is very good if you are using for the sample for the research application purpose. And if you are, but it has some disadvantage that it is very slow and it is less tissue penetrating. So if you are not in a hurry and sample is not urgent, then you can use this type of reagent, but this type of reagent mainly used for the research application. So usually the concentration of 40% is most commonly used in case of chelating agent if you are using EDTA. Sorry. And in the end point of decalcification is the most important points while using the different types of reagent, because if you don't know what is the actual end point of the decalcification reagent, then it may cause either the over decalcification or the incomplete decalcification. In that case, what will happen? If it is over decalcified, then it will spoil the tissue samples morphology and it will hamper the problem of the nuclear staining. So if the tissue is not completely decalcified, in that case what will happen? Your knife will be spoiled, because calcium is hard material and salts, it will spoil the blade. That's why you should know very careful about the end point of the decalcification reagent. And there are various methods of the to determine the decalcification. So X-ray and physical method and the chemical method. X-ray method is there, but it is very costly, so it is not commonly used, but it will find very good results. Whether it is single calcium molecule also there, you can identify. It is very costly, so it is not commonly used. Another case is physical test. Physical tests are there, just you are based on the texture of the tissue, inserting the scalpel pin, you can identify the decalcification points here, but this is not a most reliable techniques. So it has to be done under the expert histopathologist. So it is also not commonly used. Another method is the chemical method. This is the very simple method, anybody can use in the lab. Just in this case, just you have to take out the 5 ml of decalcified fluid from the container where the specimen was kept, and you have to mix with the 5% of the ammonium formate and ammonium oxalate and ammonium hydroxide solution, and you have to mix carefully, and you have to keep overnight incubation. If you are getting any precipitate, it's mean that the complete calcium has not been removed, then it require more time in case of decalcification fluid. So like that, this is very simple test to know the end point of the decalcification points. So after the decalcification, last step is the neutralization, because you are using some strong acid and weak acid that needs to be removed. This is called the neutralization. So you have to use under the running water, then all the acid will be removed. Now your sample is ready for the fixation and further steps of the tissue processing. Next is the fixation. Fixation, it is the first and foundation steps of the tissue processing steps. So results of your further subsequent steps, it depend upon these steps only. So you should be very careful about these steps, and main goal of the tissue fixation is that it maintain the tissue structure morphology as it was taken from the body. So it will maintain the original form of the tissue. Moreover, it will also stop the bacterial activity here and inhibit the autolytic activity. This is the most common main application of the fixation step, and moreover it also acts as a mordant, so it will induce the optical contrast and good pictures will come while using the microscope. And last application of this fixation is that it make the cell insensitive to the hypertonic and hypertonic solution. So what will happen, so if we are keeping the solution in some harassed condition, so morphology of the tissue will not change. So moreover, the main goal of fixation is to make the tissue able to the further step of the processing, because for the next step is very harassed and using the high temperature and agitation. So it will harden the tissue and make the resistant to the further damage if happen in further steps. Now there are various types of fixation methods are there: physical method, chemical method, and cross-linkage method. Physical method, you are using some heating or microwaving, and chemical you are using some alcohol. But most commonly used fixation is there the cross-linkage based. So in that case, formalin is most commonly used. So I will discuss about some formalin, how its work in the tissue fixation. Actually, if you are using for formalin, you should use at the 10% of its concentration of the formalin. In the market, the formaldehyde is present as a solution form in 40%, and you have to make the 10%. So finally, your concentration of formaldehyde comes under the 4% of formaldehyde. Now this formaldehyde, how it will work? Actually, it make a cross link between the lysine and glutamine residue of protein residue, and it will make a meshwork, and in this meshwork protein get stabilized and it will maintain the structure of the tissue. And moreover, the calcium, then lipid molecule and nucleic acid molecule and some sugar lipid will trap inside the protein meshwork, and it will maintain the structure of the tissue in the proper shape, and it will not change. That's the role of the formalin in maintaining the shape and rigidity of the tissue. And some other fixes also there is depend upon the types of tissue and your requirement for which purpose you are using. If you are using for microscopy purpose, electron microscopy, then you choose glutaraldehyde. If you are using some special type of tissue like nervous tissue, then you have to use osmium tetroxide. Like if you are using fatty acid, then you have to use osmic acid. So depend upon your requirement and types of tissue, you can use the different types of specialized fixation reagent. But if you don't know about the tissue, then you can use formalin at 10% of the concentration. That's why formalin is also called a routine fixative. Then after fixation, you need to remove the water from the tissue, because as you know that our main goal is to provide the rigidity to the tissue using the wax sample, and directly if water and any aqueous solution are present in case of tissue, then wax cannot be infiltrated. So you need to remove the any aqueous solution or water from the tissue, then only your wax can be infiltrated inside the tissue, and it will get the tissue embedded. So dehydration, it is the removal of the water from the tissue, and finally the places will be occupied by the dehydrating reagent. So in this case, mostly alcohol is most commonly used as a dehydrating reagent. Now you know alcohol is a dehydrating reagent it is a very strong hydrophilic molecule, and it is polar, and it will make a bonds with the water with the help of hydrogen bonds, and it will remove the water. So like that it will remove the water, and finally the tissue space will be occupied by the alcohol. And you should be careful here also, you should not use directly 100% of alcohol. So you should be process in increasing concentration of alcohol. So you should start the concentration of alcohol from 70%, then 80%, 90%, and finally you process under the 100% of alcohol, because if you suddenly use the 100% of alcohol, it will distort the structure of the tissue. So always you should proceed slowly manner and under the increasing concentration of alcohol. And the duration of the incubation is depend upon the types of tissue, which type of fixative we have used. So it depends. So you need to optimize before further steps. Finally, after the dehydration, you need another step is clearing. Clearing means what? You have to, finally you should, don't forget, you should use always wax. So now still water removed, but still dehydrating reagents are there. So you use dehydrating reagent, and which you are using here is alcohol. It is not miscible or mixable with the wax. So again you need to remove the dehydrating reagent here. So you need to use, or you can find some reagent which is mixable or mixable with the both mean it is mixable with the alcohol as well as to the wax. So here the Xylene play a major role here. So what Xylene will do, it is mixable with the both alcohol as well as the wax reagent. So first what it will do, first it will remove the dehydrating reagent from the tissue, and finally it will replaced by the wax. So in clearing reagent, what is there, it is a removal of the dehydrating reagent from the tissue, and finally the Xylene will be placed inside the tissue. So this is called the clearing reagent. And what is the meaning of clearing? Because what is the meaning of clearing is that the clearing, it gives the transparency to the tissue and optical clarity. That's why its name is called clearing. Like Xylene is the most commonly used, it will give the transparency to the tissue. That's why its name is this steps is called clearing agent. And this clearing agents, because why it is called clearing agent, because it also help in the to knowing the end point of the dehydration. Because if dehydration is not done properly, then you will not get the tissue proper transparent. So that's why it is a very important steps. So just Xylene is the most commonly used. Other than Xylene, you can use benzene, chloroform, other reagent as a clearing reagent. Next step is the infiltration and impregnation of the tissue. In that case, what you will do, you have to use the paraffin as an infiltration reagent. So after clearing the tissue, after clearing the tissue need to be transferred into the molten paraffin for infiltration and impregnation. So in that case, you have to use the molten paraffin as a infiltration reagent. So what will happen, this paraffin will infiltrate to the tissue at the molten stage, and finally it will solidified and it will get fixed inside the tissue. So this is called the infiltration and impregnation of the tissue. In that case, what will happen, this impregnated tissue will provide the support to the tissue, and it will help in the dissection sectioning during the microtomy. And mostly you are using the paraffin as a infiltration reagent. Now we come to the last step of the tissue processing steps. This is called the embedding or blocking out. Embedding mean what? Just you are fixing the tissue in the some solid material, because directly you cannot use the tissue in the microtome. So first what will happen, you have to fix the tissue in the embedding material. You are using mostly here what? Molten paraffin in a solidified form. So just you have to fix the tissue in the paraffin, so it will give the block shape, and then block you can fix under the microtome, and you can easily cut. So because directly you cannot use the this reagent as a tissue as a sectioning. So these are some apparatus used for the embedding. So mostly you are using some stainless mold type structure and paraffin for embedding, and you are using a plastic embedding cassette also for the embedding process. So these are the process of the embedding. First you have to take the molten wax in the mold, then you put the tissue, finally you have to allow to cool. After when it is solidified, then again you have to put some extra molten paraffin on the tissue, but you have to, before putting the top of wax, you have to add the tissue cassette. Now again you have to put the under the cold condition, so it will solidify. Finally you need to remove the tissue cassette, and finally your block is ready for the microtomy. So that's all about the tissue processing steps. So in the next lecture I will discuss about the microtomy. So what is the process of microtomy and different types of microtome I will discuss. Thank you.